56 years ago when I was a high school student. This was me a few years later, traveling through Asia. And I didn't visit the Ukraine then, I visited it before, but in the trip to Ukraine, I spent time in the lovely city of Kharkiv, and then camping across Ukraine and to Odessa. It was a wonderful time. And so I'm now an old man, but I've had many adventures. And that's what I like to recall for you today, balancing the scientific studies of my career, plus the personal interests in the people that made the science happen. So my interest initially was in studying cholera, a horrible diarrheal disease that affects children and kills thousands of children a year. I also was interested in malaria, but that work took a long time to get to. So when I was a student, I worked in a laboratory. This was back in the, excuse me, in the 1970s. We had long hair and beards, and the young people in our laboratory were from all over the world, from Taiwan, from Israel, from Europe, from Poland, from Spain. And we had a wonderful time working on our science. And it brought us together in a special way. These two young men laughing. It was interesting because on the left is Naji Sayhoun, a Palestinian scientist, graduate of American University of Beirut. And on the right is Marvin Ira Siegel, son of an Orthodox rabbi from Brooklyn, New York. And while they initially were hesitant because of the backgrounds were antagonistic, they became the best of friends as scientists and stayed friends the rest of their lives. There's something special about science that brings us together. So when I started my own laboratory, this was when I was in my mid-30s. I had a wonderful technical assistant, Barb Smith, shown her picture, and we were studying red blood cells. And we discovered a new protein. If you look over at the far right, I have a pointer, I hope you can see that. That is a protein that we were able to purify that had not been seen before and really drew our interest. So we decided to clone it out and study it. And we found the red cell had a protein in the membrane, which was similar to the proteins identified in the lens of eye, the brains of insects, bacteria, and plants. So it was a protein that was interesting biochemically, but we had no function for it. And there is when the magic of science needs the magic of human relations. On a camping trip with our children to Florida, we stopped in Chapel Hill, North Carolina, where I visited a former professor, and I told him about this new protein in red cells. We thought it might be important. It's a membrane protein, we felt, possibly a channel, but we didn't know a channel for what. And it was this dear professor, John Parker, who put it together. He thought, membrane protein channel in red cells. It was also present in renal tubules. He said, have you considered this might be the long sought water channel, the channel that allows water to cross cell membranes rapidly in some tissues, like red cells, that protects others, like the surface of our skin. Water is ubiquitous in biology. And at equilibrium, water is tranquil as this Arctic stream. But when water is in disequilibrium in our bodies, it's a very, very violent torrent. And this is seen in human disease. Brain water biology determines if we have normal brain function or hydrocephalus. Tears, sweat, saliva, there are many biological fluids, and they all have a special capacity to move water, but it was not known how the water moved. So it was John's suggestion that we study this further, and we did. Now all, pardon me, all membranes have a finite degree of water permeability by simple diffusion. On the left, you can see water goes into and out of the cells. But on the right, You can see that specialized water channels were believed to exist in special tissues like red cells, renal tubules, and plant tissues. We tested the function of the new protein, collaborating with a dear colleague at Johns Hopkins, Bill Gugino. And we tested this in frog eggs, injecting the oocytes, the frog eggs, with buffer alone on the left, or buffer with complementary RNA for the new protein on the right. And the oocytes express the protein, but they look normal. However, this is at isotonic conditions. When the transfer was done to distilled water, the test oocyte has swollen and exploded, and this produced much jubilation in the laboratory. Shown here is a photo of my postdoctoral fellow, Gregory Preston, and he was so excited. We were celebrating. I took his photograph 3 years after the discovery of the water channels, and he was still celebrating. I communicated with Greg just yesterday about this lecture, and he sends his greetings and shares his joy for science. But the function was not altogether clear. The protein was solved, the structure of the protein was solved in collaboration with Andreas Engel in Switzerland, Yoshinori Fujiyoshi in Kyoto, Japan. And it shows that the structure allows the passage of a single water molecule, shown on the left with space filling representation. This is work of David Cosino, now a Harvard faculty member. He was a student with us. So water crosses the cell membrane at the surface of our cells, entering the cells through the aquaporin water channel, and it will also leave the cells through the aquaporin water channel, depending on the osmotic gradient. So this is the canal for water, and we termed it aquaporin, and the name is now the official name, and our first protein is now known as aquaporin 1. Now, what does aquaporin 1 do in kidney? Because kidney is the organ of greatest significance in terms of water transport. In a given day, we will filter 200 liters of plasma and reabsorb 99% of the water in our kidneys. And to establish the sites of expression, we teamed up with Søren Nielsen, a young scientist who had just finished his medical and research studies in Denmark. I communicated with Søren yesterday. He's doing well and sends his greetings. And his work as a very young scientist established the location of the aquaporin 1 protein in the kidney. And the kidney is formed of 1 million nephron units. This is where filtration occurs in the glomerulus. And in light green, I've highlighted the water permeable parts of the proximal nephron. And that's exactly where aquaporin 1 is expressed. You can see the staining of a tubule. The primary urine passes through and is surrounded by a brush border with aquaporin 1 in both the apical surface and the lateral and basal surfaces. The collecting duct, the terminal part of our kidney, where the final concentration occurs, has no aquaporin 1. It turns out to have another protein. And so it explains how water enters from the primary urine into in the kidney, crossing the proximal tubules by entering through aquaporin 1 in the surface and leaving aquaporin 1. So water can pass right through the cells. Now, does this have clinical significance? And the answer is yes. Our colleague Landon King, former postdoctoral fellow shown here, with 2 French ladies. The older one was a retired school teacher in France, and with her permission, I show her photograph. She turns out to have a genetic defect which leads to, prevents the synthesis of aquaporin 1. And the clinical phenotype is that she cannot fully concentrate her urine. And a typical individual, after overnight thirsting, and when we go to bed and sleep 7 or 8 hours, we're thirsting at the end. We concentrate our urine. And all normals can concentrate up to 1,000 milliosmolar or beyond. The null phenotype, aquaporin null, can't concentrate beyond about 420 milliosmolar. So they have a mild form of nephrogenic diabetes insipidus. It's quite rare, but it shows the function of the aquaporin 1 protein. Now, based on the discovery of aquaporin 1, we and others have identified numerous aquaporins in many species. It's believed that all life forms have an aquaporin or an aquaglyceroporin, which is a member of the family which is permeated by water plus glycerol. And I'll tell you just briefly about each of these, highlighting the young people who did the experiments. So aquaporin 2 was identified from the collecting ducts. And this is where the final concentration of urine occurs. And the mechanism is by inserting the protein in response to antidiuretic hormone vasopressin. Another aquaporin from lens was identified by Masato Yasui, a young Japanese scientist who had done his PhD training in Sweden and came to Johns Hopkins for several years. And he established that mutations in the lens homolog, aquaporin 0, causes cataracts in small children. Aquaporin 4, the water channel of the brain, has been looked at extensively by a number of investigators, including our colleague Ole Petter Ottersen, shown here. He was a young Norwegian neuroscientist, and he's now the rector of Karolinska Institute, the medical university in Stockholm that awards the Nobel Prize. Aquaporin 4 in brain is very important. And it was Ole Petter's prize student who established the pathological significance of the aquaporin 4. His name is Mahmoud Amiri Moghadam, and you'll recognize that's not a typical Norwegian name. Mahmoud began his scientific career as a refugee in a camp in Pakistan. He was adopted by the Norwegian Social Services and came to Oslo, where he was a brilliant student, brilliant medical student, now a leading European neuroscientist. And what he has established is that after a defining brain injury due to artery occlusion in mice, the normal mice sustain a significant amount of edema and infarct. You can see the large infarcted area. When the same artery is occluded and then reperfused in mice that have mutations in the gene encoding the bracket that shows aquaporin 4 in place, those mice look normal, but they're spared from the severe damage of the arterial occlusion. This gives us great hope that we may finally have a medicine to prevent or treat brain edema, because it's very severe and it's very common after head injury, automobile accident, a closed head injury in athletics, infarct, a stroke, or a tumor. And many of us have had family members who've had brain edema from one of these causes. And shown here is a very personal example. This is my dear wife, Mary, a few years ago, when she developed over the course of a few weeks fatigue, headache, and some minor visual impairment. And her physician sent her for a CT and saw a huge frontal lobe brain tumor. By MRI exam, on the upper left, you can see the normal brains surrounding this brain tumor. And when the MRI images are weighted to the density of water, you can see that the tumor is surrounded by a large amount of brain edema. Her life was saved by a neurosurgeon who removed the tumor, but the edema took weeks to dissipate. Here's Mary after surgery. The headaches were improved, and here she is back in her normal state. Another member of the Aquaporin family, Aquaporin 5, was cloned and studied by Surabhi Rain, a wonderful postdoctoral fellow from India who worked in our laboratory and is now a professor at Syracuse University in New York. Aquaporin 5 is present in salivary glands, lacrimal glands, and sweat glands, and it's the mechanism for the release of water into sweat. This is a very simple assay looking at a normal mouse on the left and a test mouse with Aquaporin 5 targeted gene disruption on the right. And as you can see, the number of functional sweat glands is greatly reduced in the knockout animal. Now, the pore diameters of the aquaporins and the aquaglyceroporins are not identical. And the structures of the residues explain why the pore size could be increased for the glycerol transporting aquaglyceroporin. The aquaglyceroporins are present in the basal levels of skin, and that's how our skin responds so well when we put glycerol-containing hand creams on dry skin. The beauty industry got into this, and their advertisement is a bit overstated. They talk about spectacular results and the Nobel Prize in chemistry. I showed this photograph to my mother, who was at the time 80 years old, and the company that markets this hydroxyl skin cream had a very vigorous advertising campaign, and my mother said she thought I was finally doing something useful. Well, I hope our work is more useful than beauty creams, but it shows that the science can go into areas that are new to the experience of the investigators and maybe even doing something useful. Aquaglyceroporins are also very important because they reside in fat tissue and in liver. In the late stages of fasting and starvation, the adipocytes in the fat tissue release glycerol and fatty acids. The glycerol release occurs through aquaglyceroporin 7, and it's taken up by the liver where through aquaglyceroporin 9 and converted to glucose. So we can maintain blood glucose levels even at the very late stages of starvation. This is work of Jen Carberry, now a young faculty member at Duke. Now, red cells have aquaglyceroporin 3, and that turns out to be very important in malaria. Work from Nirbi Kumar, a scientist from India who's now a professor at George Washington University, and Dominique Promeneur, a French scientist who's now working in West Africa. And as you can see, the red cells shown here have been infected with the malaria parasite. Here's a single parasite. When it divides inside the cell and eats the hemoglobin, it turns into a schizon with many malaria parasites that bursts and causes horrific fevers and tragic events, even death for the children that suffer from this. And the mosquitoes that transmit malaria also have aquaporins to maintain osmotic fluid balance and to prevent them from having too much water on board and reduces their ability to fly after taking a mosquito bite. Aquaporins are also present in plant tissues, and this lovely demonstration by Ralph Kaldenhoff from Würzburg, Germany, shows that the Arabidopsis, this is a small mustard plant, has leafy foliage and is maintained by drawing water from this arborization of rootlets. Kaldenhoff genetically reduced the expression of the aquaporins in the rootlets, and the plant compensated. When it couldn't make as many aquaporins, it would send out more rootlets to compensate. Plants compete for water also. And this was a early morning photograph taken quite a few years ago of my laboratory at the morning I received a call from Stockholm informing me that I would share the Nobel Prize in chemistry for our study of water channels, discovery and study of water channels. And the young people in this photograph originate in 8 different countries. Science is not the possession of any one individual or one university or one country. It belongs to us all. And the joy of having international postdocs and students in the lab has been one of the great adventures of my life. But it caused me to reconsider our efforts. And shown here after the Nobel ceremony, I'm surrounded by my wife, my children, and we had at that point decided we were going to move into malaria to see if the aquaporins had an important role in malaria. Now, malaria used to be worldwide. It's still very prevalent in the poor parts of the world, most notably sub-Saharan Africa. And I'm going to tell you briefly about our work in Africa on malaria. And we have an active research program in Zambia. And to do the research in rural Africa, you need the permission of the tribal leaders. Very, very gifted individuals. This is Chief Macha, who has a group in southern Zambia that we were studying because there was a hospital there with an American doctor. We could make a research unit. And here he is. This is the American doctor, excuse me, Dr. Philip Tuma, taking care of the patients with malaria. And of course, malaria is a terrible disease because it affects the poorest families and the young children in those poor families. This is a youngster who was brought into the hospital, the field hospital, in near death, in coma. His life was saved by Dr. Tuma, but as you notice, his vision he's been blinded by the malaria. His visual cortex in the brain was destroyed. So he survived, but he was never able to see again. So in addition to the many deaths, and it's believed there were more than 600,000 people died of malaria last year. More than 80% of them are children in sub-Saharan Africa. And because of the gift of Michael Bloomberg and the Bloomberg Philanthropies, our research institute is doing field studies. And here I am with our African scientific leaders. And I'll come back to tell you about one in particular, this gentleman in the blue shirt, who is a Zimbabwean scientist that has taken a leadership position. And coincidental with our study of malaria, the treatment was effective in screening all the relatives of the children and treating them. The burden of the disease has declined. So the regional hospitals and medical clinics, which would normally have 60 to 100 deaths per year, have improved such that over the last 3 years, there have been no deaths. We know what will happen if we stop our efforts, however, the malaria will come back. And these people in the outback are probably the last innocent people in the world. They're so kind, they lead very meager lives, but I think when the crops are in, they're doing pretty well. And here's one of our field workers, Harry Hamapumbu, who's doing a blood analysis from a farmer whose children had malaria to see if he was a carrier. And I'm going to go rapidly through the photographs now because they basically underscore the message that these people lead very simple lives and they need they need our help. I think we're obliged to do so. Using methods to farm that were discarded in modern Europe and the United States more than a century ago, they support their families. And the children work too. The older children take care of the younger children. They bring the maize to the village to grind it into cornmeal. And they know how to have fun. They take their children very seriously and contribute to their education. And I think this is going to be bring a bright future to Africa. We also worked in the Democratic Republic of Congo, which is more challenging. The government in Congo has been in disarray for a long time. The regime of Mobutu Sese Seko was horrible, with gross corruption and bankruptcy of the country, but the people are wonderful. And shown here is my colleague Bill Moss from Johns Hopkins lecturing to the Congolese medical students. Now, our work in Congo is in the remote areas, and we were dependent on the missionary pilots to fly us up to the sites that we studied because there were no paved roads. And here we are with some of the village leaders in a village in Katanga province who were devoted to the well-being of their people and particularly the children. These children have very modest lives, and most of them will get malaria every year. So this is a burden that doesn't need to occur. And we think our efforts are going to help reduce and eliminate malaria. It's also a place in the world where there's been much turbulence with the death of Doug Hammarskjöld, the Secretary General of the United Nations, occurring while he was in Congo, attempting to broker peace. We're also working in Zimbabwe, which is across the river from Zambia. We refer to them as Zim and Zam, beautiful countries, but with a dictatorship, which is very autocratic. This is Robert Mugabe, who was the liberator of Zimbabwe from white rule, but he retained office for 35 years and became increasingly corrupt. And the economy collapsed and malaria returned to Zimbabwe. How can we do some research in a country with an autocratic leader? And the answer is sometimes the diplomacy. We are partnering with an institution in Harare, which is directed by this lady, Shungu Munyati, who was a life scientist. And she also turns out to be the niece of Robert Mugabe. So sometimes the collaborations require a little compromise of our political ambitions, but we're not politicians, we're scientists. It gives us the opportunity to do the work. And Zimbabwe is a beautiful country. Here's the rich tea country on the border with Mozambique, some of the workers. But the people lead very simple lives. It's basically their lives are like camping. They rely upon the streams for the water supply, for washing, for cooking, for religious purposes. Here there are some Zimbabwean children on their way to school. And this photo is an elderly Zimbabwean man about my age who shared with me that his ambition was to see to it that malaria was eliminated from Zimbabwe forever, so his children and grandchildren would not have to suffer from it. They have wonderful nursing staff at their health centers, but they have problems with the stable and reliable sources of medicines. And during the malaria season, when the mosquitoes are out, the rainy season, the clinics are jammed every day with young mothers and grandmothers bringing the small children in to be checked for malaria. So shown here is my friend Sungano, a Zimbabwean child who made his way to Oxford University, earned his PhD, and then returned to Zambia first, where he worked with us, and now Zimbabwe, where he's the Dean of Life Sciences at the Africa University in Eastern Zimbabwe. And he's committed his life to eliminating malaria. And we're so proud to have him as our colleague. So in closing, there's a word in Mandarin for crisis. It's Wei Ji. Wei means a time of danger. Ji means a time of opportunity. And I think the medical sciences are just that. Of course, there are dangers that are biological or natural that we have to deal with. And then there are the dangers of warfare, which we're so sorry to follow the tragedy that is occurring in your country, Ukraine. But the opportunity of pulling together through science and using it for peaceful purposes remains. And with that, I'd like to thank you for the privilege of talking to you. If you see this by tape, I wish you the best. And those who are present, if they have questions, I'll do my best to answer them. Thank you so much.
Thank you, Professor, for your presentation very much. So we have many questions. First of all, I will ask some questions related to your topic, to your talk, and then some questions related to motivation and some overall part. So the first question is from Sergei. How did your life change when you became a Nobel laureate?
Well, it became increasingly difficult to maintain privacy. Having some attention is wonderful, but having constant attention is not something that many people would want to do. The expectations that I would do something marvelous on every occasion is hard to live up to. But I think in terms of the public recognition of science, it was a very good event because I was able to thank the public who paid their taxes, and the tax money supported the research in the laboratories, as well as the donations from foundations and individuals. Michael Bloomberg, former mayor of New York, was a major donor to our program. So it's both good and it's both difficult. The challenge is to maintain our direction and keep working with so many people that are asking for favors and invitations, and it's almost too much for one person. But in the end, to be part of science in a historical way is something I'm very gratified.
Okay, the next question is from Anastasia. Is there a disease in which a person has too much aquaporins? If so, is there a treatment?
Very good question. In fact, our bodies regulate the amount of expression depending on our hydration status. In the kidney, the collecting duct is the terminal part where water is reabsorbed. And if an individual is thirsted, the individual will increase the bioavailability of the Aquaporin 2 in collecting duct by trafficking it to the cell surface and increasing the number of copies. So our antidiuretic state has an excess of aquaporins. When we are rehydrated, the protein is reinternalized and it's also shed into the urine through vesiculation, because it would cause us to reabsorb too much fluid. And this is found in states like congestive heart failure and even some pregnancy, where fluid retention is problematic. And this is due to the excess expression of Aquaporin 2. Deficiency of Aquaporin 2 invariably results in polyuria, and the problem that some small children have with bedwetting is because they have underexpressed the Aquaporin 2, and they will grow out of it. their parents are, of course, very worried. But bedwetting is not considered a serious disease, but it is a manifestation of insufficient number of aquaporin 2 proteins in kidney. I hope I answered the question.
Okay, the next question is from Yelizaveta. How did aquaporins evolve in living organisms?
How did aquaporins evolve in living organisms?
Yeah.
This is a profound question. It doesn't have a simple answer. The protein has an internal repeat. It has 6 bilayer spanning domains. So the front half and the back half of the molecule are similar, but they're oriented in the opposite direction. So we think there was a progenitor molecule with 3 bilayer spans, which duplicated, and that folding allowed water permeation to occur. But it's been found in all life forms, including archaea, the third kingdom of bacteria, and it's something that probably occurred very early in evolution. This is conserved in all life species. It's not always essential for life, however. Studying yeast, Saccharomyces, in laboratories, the yeast are washed with fresh water, and they're selected for yeast which have mutations in the aquaporins. So we can screen for them artificially, and the yeast do fine in the laboratory, but they would probably not survive in the wild. So we don't know more about the evolution, I'm sorry.
Okay, the next question is from Sviatoslav. If a person does not drink enough water or has dehydration, changes in aquaporins, does it make changes in aquaporins or does not affect their work in any way?
It does. I mentioned a few minutes ago the aquaporin 2 in the collecting duct. And so if someone is thirsted or becomes dehydrated from excess exercise in hot weather, the kidney shifts into the antidiuretic state. So aquaporin 2 is present in the surface membranes, so water can be reabsorbed from the primary urine back into the bloodstream. If an individual had what we call syndrome of inappropriate ADH, inappropriate vasopressin, they would be retaining water and it could become toxic. So the hydration status has a lot to do with the level of expression of both aquaporin 1 and aquaporin 2 in kidney.
Okay, the next question is, can cause water retention in the body or rapid weight gain, for example, obesity, failure of aquaporins?
Yes, but fluid retention, this is a major problem in clinical medicine. Congestive heart failure. An individual sustains a heart infarct and the pumping action of the heart is reduced. The kidneys sense underperfusion and they release they respond to vasopressin that is released from the brain, and then they will retain fluid. And it's the fluid retention that often kills the patients. So fluid retention is most definitely related to excess expression of aquaporin 2. And the opposite for polyuria, as I mentioned, bedwetting or other forms of polyuria. These are often due to insufficient number of aquaporin 2 proteins in the collecting duct.
Okay, the next question is whether such a protein can be synthesized in vitro.
Well, the protein biosynthesis is complex because it's a membrane spanning protein. And we can certainly express it with yeast and bacteria, and they have their own aquaporins, but that's great for research purposes. It's not a problem. It is as many membrane proteins. If you have the proper complementary RNA, it will be translated and expressed into the bilayer as it is translated.
Okay, now we move on to the part about motivational questions. So the question from Mikola: what are your favorite items about being a scientist?
I like the opportunity of working with young people, because science is for young people. When I was young and working at the bench, that was a wonderful time in my life, and I made lifelong friendships. So I think the social aspects of science is really wonderful. And the chance to collaborate with scientists all over the world. I have friends in countries that our politicians could never enter. North Korea, and Iran, and Cuba. I think science is a special endeavor, and we're not selling anything. We're giving away knowledge. It's reassuring, and I get much encouragement from people whenever I meet them, and they learn I'm a Nobel scientist directing a malaria research unit. They congratulate me and thank me. I don't feel like a hero, but they consider that heroic.
Okay, the next question is, what is the most important item to become a successful scientist?
I think you have to have the ability to withstand failure and experience a lot of disappointing experiments, but with care and some luck, you sometimes find something unexpected, which is very important. So I think if someone is too impatient, they will give up. Oftentimes, perseverance makes all the difference. I think also maintaining dialogues with other scientists. In our case, we discovered a protein. We didn't know what it did. We didn't know it was important. And it was a conversation with a former professor that changed everything. So I think being open and honest are also very helpful. Honesty is essential. If a scientist is not honest, then the work is no longer regarded as reliable.
Okay, the next question is, how did you overcome challenges you faced? What do you usually do to not give up?
Well, I'm fortunate. I have a wonderful wife and children and colleagues, and they all encouraged me. My wife in particular, we had a very difficult time when one of our children was severely ill, and I had to make a decision whether to continue science or quit the science and take a job which had a guaranteed salary. But she encouraged me, and it stayed in science, and it turned out pretty well. But without her encouragement, I probably would have given up.
Okay, the next question is, what is your current project? What do you plan to research, to investigate in the nearest future?
Well, I'm 73 years old and I have Parkinson's. My dexterity is not good. My project is not at the bench any longer. The technologies change very rapidly. So the younger scientists should lead that. What I hope to achieve is to use occasions like this morning to use science as a form of diplomacy, to create friendships amongst countries. I have one regret, in that it was about 10 years ago when I was in Warsaw. I was inducted into the Polish National Academy of Medicine, and the president of a major university in Kyiv and the president of a major university in Odessa both invited me to come to their campuses. And I was just too busy. I couldn't fit it in that year and lost track. And now I'm very sorry that I didn't visit Kyiv and Odessa to visit those universities. There are wonderful people at both of those universities and the other universities. I think Ukraine has this long tradition of excellence in science. Ervin Shargaf, who recognized the the base pairing that goes into DNA, was Ukrainian-born. Theodosius Dobzhansky, a developmental biologist, was Ukrainian-born. Solomon Waxman, who discovered streptomycin, the first drug against TB and won a Nobel Prize, was from Ukraine. So I look forward to living a long life and visiting those universities, in Ukraine in particular.
Yeah, we are waiting for you and waiting for your lecture in Kyiv. And the last question is, what you can advise to young scientists, to school students and students now?
Well, right now, things are very difficult in Ukraine. So be aware of what you need to do to keep safe and to maintain safety amongst your family members and your friends and neighbors. The world is watching and hoping the outcome will be positive. And once that is solved, I think the important thing is to listen carefully in your scientific classes and find out what really interests you. Because when you find a project that strikes you in your heart, you will work on that day and night. And sometimes the outcome is surprising and extremely important. So don't give up.
Thank you very much for your presentation, for joining our project and wishing our students all the best. Thank you very much. And we are waiting for you to visit Kyiv one day.
Well, my best wishes to all, and thank you to Dr. Kudryavets, who arranged this. So long.
Thank you very much. Шановні учасники, дякуємо всім за те, що приєднались до нас до останньої квітневої лекції. 2 травня ми розпочинаємо наш травневий цикл, тому реєструйтесь. У нас дуже насичений графік. Будь ласка, заходьте на наш оновлений сайт mango.ua і чекаємо на вас у травні на наших лекціях. Всім гарного дня, на все добре, до побачення і до зустрічі. Thank you very much again. Goodbye, have a nice day.
Thank you. Farewell.